Good afternoon, everyone. Thank you for joining the Habitat and Ecosystem session. We will start now. Yes. Thank you for joining the Habitat and Ecosystem session. We're going to start now. The first presentation will be recorded and online, and you will be able to ask questions at the end. So thank you in advance for your attention, and to have a wonderful session.
Hello, my name is Amanda Catron, and I am a program specialist at the University Corporation of Atmospheric Research. I am an affiliate at the U.S. National Oceanic and Atmospheric Administration's Climate Program Office, specifically situated in the Adaptation Sciences Program. Today, I am going to talk about the NOAA Blue Carbon Inventory Project, and which I am a project coordinator for, and how this project enhances global coastal resilience through blue carbon. The NOAA Blue Carbon Inventory Project is a multi-agency project led by the NOAA's Climate Program Office in partnership with the U.S. Department of State. This project is also part of the larger Department of State Transparency Accelerator for Greenhouse Gas Inventories effort, which involves additional components with partner agencies such as the U.S. Environmental Protection Agency. Along with other U.S. agencies, we also partner closely with other parts of NOAA, such as the Marine Protected Area Center or the MPA Center, and external partners such as Sylvestrum Climate Associates, the Smithsonian Environmental Research Center, and the Ocean Foundation. This project has two primary components, with coastal blue carbon ecosystems at the core. The first is integrating blue carbon in national greenhouse gas inventories, and the second is supporting conservation and management of blue carbon habitats. Inventorying blue carbon ecosystems at a national and international scale is fundamental to conservation, restoration, and effective management of blue carbon ecosystems. However, additional capacity and resources for inventorying are often necessary in order for countries to meet international emissions reporting guidelines. The two components of the Blue Carbon Inventory Project work together to enhance the capacity of partner countries to integrate coastal wetlands into their national greenhouse gas inventories and enhance their capacity to manage these ecosystems, which then further realizes the co-benefits for adaptation and resilience. This slide shows a diagram of our project workflow, and I'm going to go a little bit more in depth on the following slides about the different steps of our process. So the first step of our process is assessing needs and prioritizing scope. We communicate with our partner countries to understand their current capacity and resources. The next steps in the process kind of go hand in hand and are building country-level awareness and capacity, addressing data and knowledge gaps, and expanding data management tools. In order to inventory coastal blue carbon ecosystems, countries must map and collect data. There are a couple different components that feed into this. So ecosystem mapping can leverage remote sensing techniques to more accurately map and monitor blue carbon ecosystems, estimate carbon stocks, and track changes over time. However, remote sensing needs to be calibrated and validated through field measurements. So once coastal blue carbon ecosystems have been mapped, field sampling must be conducted across ecosystem types and regions to quantify the amount of carbon in these systems. The methods for assessing carbon stocks vary by the part of the ecosystem that is being considered. This means that the method for evaluating the carbon stored in biomass or living material above ground is different than below ground. And finally, effective stewardship of data is key to building a stable, robust library of information from which to draw continuous insight. So addressing limitations in data management infrastructure can help strengthen in-country capacity to support blue carbon projects and inform decision-making at a community, regional, and national level. In-country data may exist, but it's often not findable or perhaps not in an accessible format. The NOAA Blue Carbon Inventory Project supports researchers, resource managers, government staff, and others in data cleaning, reporting, management, and analysis through training and direct technical support. This is done through the Coastal Carbon Network, which maintains the largest data library of carbon data from coastal wetland ecosystems around the world. So looking back at our workflow, we're now going to focus on step 5, which is developing national inventories. It's important to integrate coastal blue carbon ecosystems into national greenhouse gas inventories, as I mentioned before, in order to enhance in-country capacity to manage these ecosystems and to further realize the co-benefits for adaptation and resilience. The 2013 Intergovernmental Panel on Climate Change, or IPCC, provided supplemental guidance to address the gap of carbon into national greenhouse gas inventories. The NOAA Blue Carbon Inventory Project provides peer-to-peer assistance and direct technical support to national governments that are responsible for national greenhouse gas inventories. This assistance is underpinned by the development of templates and worksheets that help make the IPCC wetland guidance more easy to understand and implement within a broader national greenhouse gas inventory process. And finally, let's discuss the outcome of our workflow, which is improved carbon and coastal management. One of the most effective ways to protect the many benefits of coastal blue carbon ecosystems is to ensure they are conserved and intentionally managed for those benefits. This is often done through the management or establishment of coastal and marine protected areas, or MPAs, or other effective conservation measures that include and manage coastal blue carbon ecosystems. The NOAA Blue Carbon Inventory Project focuses its technical assistance and capacity sharing efforts towards MPAs and MPA networks. In order to effectively manage coastal blue carbon ecosystems within protected areas, the staff and managers of those protected areas map and mitigate potential threats. In some cases, conserving and protecting a blue carbon ecosystem may not be enough to ensure it is meeting its potential to contribute to climate change mitigation, coastal resilience, economic prosperity, and local community activities. When an ecosystem is too degraded, restoration is often considered. Ultimately, consistent, accurate measurements and ecosystem monitoring underpins conservation and protection activities, threat mapping and mitigation, and restoration for coastal blue carbon. Now I'm going to move on to some examples of the work that the NOAA Blue Carbon Inventory Project has done within 2 of our partner countries. So the first that I'm going to focus on is Costa Rica. So in partnership with the government of Costa Rica, the NOAA Blue Carbon Inventory Project led a 3-part in-country training series on blue carbon for relevant government staff and partners. This included a workshop in March of 2022 on developing inventory worksheets and identifying priorities related to the sustainable management of blue carbon ecosystems. Then in September 2022, the project led a workshop for Costa Rican protected area managers and National Wetlands Program staff. And finally, a workshop in February 2023 on the integration of coastal blue carbon ecosystems into national greenhouse gas inventories. This was the NOAA Blue Carbon Inventory Project's first major engagement, and it resulted in enhanced interest in policy and capacity around blue carbon ecosystems in Costa Rica. It also enabled opportunities for exchange of ideas and knowledge and successful identification of joint work leaders at different levels within the Costa Rican government. The next example I will briefly cover is NOAA Blue Carbon Inventory Project's partnership with Ghana. In the fall of 2023, the project held 2 workshops on inventorying and managing coastal blue carbon ecosystems in Accra, Ghana. The first training focused on field data collection for coastal blue carbon ecosystems, and the second focused on coastal blue carbon and marine and coastal management. Together, these trainings engaged 15 Ghanaian institutions, including government agencies, universities, and community organizations. Through these workshops, the project initiated a technical exchange that bolstered the government of Ghana's ability to inventory, protect, and restore blue carbon ecosystems, contributing to national and global climate change and conservation goals. The in-country networks established through the workshops continue to be active on blue carbon issues today. The engagement was a catalyst for collaboration in Ghana and fostered increased regional and international engagement. It also created awareness and use of existing data and supported ongoing capacity sharing and technical assistance. The NOAA Blue Carbon Inventory Project is continuing to scope opportunities for further partnerships. Besides the 2 partnerships I just covered, we are currently engaging with Indonesia, the Philippines, Palau, and Senegal. I have put on this slide countries that are eligible for technical assistance under the Blue Carbon Inventory Project, authorized by the Department of State. As the Blue Carbon Inventory Project has expanded, we were able to add more countries to this list and are excited to continue to expand our reach. So thank you all for listening today. Please feel free to scan the QR code on the screen in order to access our NOAA Blue Carbon Inventory Project 2-pager. If you'd like additional information or may be interested in collaboration, please do not hesitate to reach out to me via email. And thank you again, and please let me know if you have any questions.
Thank you, Amanda. So we have time for questions for Amanda, if anyone. Yes, Emma? I don't know who has.
Thank you very much. Is she with us online to ask a question? Yes. If so, excellent. Yes. Okay, this is Emma Doyle.
Yes, I'm here. Thank you.
Emma Doyle from MPA Connect, which is GCFI with NOAA, helping with MPA capacity building. And one of the big topics that we work on is sustainable financing. So people say you shouldn't really talk about money, but my question is on money. And I'm wondering, has the inventory helped to open up blue carbon financing opportunities? Are there any links or experience you can share in relation to that?
Yes, definitely. It is not necessarily covered in the scope of our project specifically, but I have seen a lot of overlap when we've been engaging with different countries and doing different partnerships. There's been a lot about blue carbon and carbon accounting and financing. So we've definitely seen a large overlap there. We're hoping in the next iteration of this project that we can expand the scope and kind of explore more on that front. Right now, that's not covered in our scope, but we're hoping that since we've seen such a large interest in this, that we're able to expand there and engage with different countries regarding that topic.
Any other question? Okay, I have one. Yeah, I find very interesting, and thank you so much for bringing the subject on blue carbon in the Caribbean and seeing how it extends globally. I was wondering how the countries are receiving the idea of integrating this blue carbon assessment in the NDCs.
I think that this has been received pretty positively so far. We're kind of a, like a demand-based project. So we do have a lot of countries that have reached out to us who have kind of sparked interest, or when we have gone to different conferences or kind of presented on this project, they have reached out to us about, collaborating with this project. So I think so far it's been pretty well received, and kind of as these different topics have grown regionally, like for example with Ghana, a lot of neighboring countries such as Senegal and other West African countries have kind of started thinking more about this idea and have wanted to collaborate with us and kind of expand on that topic. So, so far I think pretty well received, and hopefully, as we do different engagements in different regions, we'll be able to kind of collaborate with others in those regions who may be interested and continue to expand from there.
Thank you. Last question, burning one? No? Thank you so much, Amanda, for joining us, and thank you for your presentation.
Thank you very much.
Next in is Fabien Blanchard, who is the regional representative of IFREMER, which is the French Organization for Ocean Science, and he's going to talk to us about the Rivière de Cayenne.
Okay, thank you, Frédéric. Good afternoon, everyone. So I am Fabien Blanchard. I used to work on fisheries sustainability in French Guiana. And today I want to tell you a story, or a short story, about the implementation of an ecosystem-based approach in French Guiana. And as we speak about ecosystem, we speak about complexity, because ecosystem, it represents several components with interactions, with various uses, with various pressures. That's why we have to involve a multidisciplinary team. And the people engaged in this work, some of them are listed here. So we have people involved in mud banks hydrodynamics, people involved in mangrove ecology, people involved in natural resources economics, and people working also on fish larvae ecology. Okay. So to introduce my talk, I thought it was relevant to explain why we move from a single species fisheries management to an ecosystem-based management. Actually, in the EU context, because French Guiana is part of Europe, the fisheries management are concerned by several policies in the EU. And there are two main contexts, two main policies. The first one, of course, is the common fishery policies. And this policy focuses on stock conservation and fleet management with the main target that is the maximum sustainable yield for each stock. That's why we call it a single species fisheries management. However, the CFP recognizes now that it is necessary to implement an ecosystem approach to fisheries because a single stock approach is not sufficient. And for example, they say that we have to avoid bycatch and discards. But apart from these two categories, it is not precise how to what is the way to implement the EIF in the European waters. So we can find, of course, tools in the literature, especially from FAO, but the CFP does not precise the way to do this. Another policy that concerns the fisheries in the European context is the Marine Strategy Framework Directive. This time, this focus on the diversity conservation, on conserving a good state of ecosystem, and also conserve the ecosystem services that are provided by it. So one could consider that this strategy framework directive is a tool for ecosystem-based fisheries management. However, we think that is not enough because the ecosystems that are concerned by fishing impact are also concerned by other activities that could have impact on the ecosystem, such as tourism, aquaculture, coastal urbanization, and so on. And biodiversity is also altered not only by fishing, but by other driving factors, such as climate change, pollution, invasive species. So this advocates for a more broad approach, a more integrated approach of fisheries management that considers the whole ecosystem and the whole pressures and activities that depend on this ecosystem. So some principles on the implementation of ecosystem-based management. There are some illustrations here, but I don't want to get into the details. I just want to underline three principles or three steps that are listed here. And first of all, of course, the question is to identify the ecosystem to manage. What is the priority? What is the most important ecosystem? What are the stakes in this ecosystem, in the society? So find the ecosystem to manage and also identify the ecosystem services that are provided by this ecosystem. Second important point is the involvement of the stakeholders, of course, and the decision maker, because they are important. They make decisions, they have impact on the management, and then they have impact on the ecosystem. They are concerned, and they have to be aware and to be informed on the functioning of the ecosystem and on the consequences of their management decision. So that's why we need to involve them. Another point, it is the last year, is the implementation of observatory. Of course, if we want to understand the functioning of the ecosystem, we have to monitor the several components of the ecosystem with indicators. and that will help to better inform the management decision about the functioning. And also it will be useful to evaluate the efficiency of the management decision that have been taken and that act on this ecosystem and the uses. So in French Guiana, what is the context and what ecosystem have been chosen for implement an ecosystem-based management? The context is the following. The coastal ecosystem is represented by around 75% of mangrove in terms of surface of the coastline, and around 25% of mudbanks and some few rocky shores. And there is a particularity in French Guiana. It's a very unstable ecosystem, a very unstable coastline, because we have migrations of mudbanks and the associated mangrove from the southeast to the northwest part of the coastline because of the currents and the waves. So this is very challenging for all the question of coastal management, land use, infrastructure building, of course, but also for conservation areas and economic development, generally speaking. So mangrove is very important, of course, and the good news is that according to the knowledge of the experts, the state of the mangrove is quite good. However, it will change in the near future because there are major pressures that are increasing, and especially two pressures, the population growth and the climate change. And as you can see here, the population, according to the projection, will double by 2050, and there will be consequences, of course, of this increase of population in terms of increased demand of food, with potentially increase of the fishery exploitation, and we don't know really the capacity of the marine ecosystem to sustain such demand without altering the biodiversity, the habitat, and avoiding overexploitation. There will be, of course, increased urbanization, increased infrastructure building, and this may have also potential impact on coastal habitats that may be very important for the life cycles of the fish that are exploited with, for example, the mangroves that act as a nursery and that may be impacted. Of course, there is increased pollutions with the land use increase, with urbanization, with agriculture, and so on. In terms of climate change, the BRGM and Météo France have shown that they will have a sea level rise, increase of temperature, and also change in the rainfall regime, and this will have also impacts on the biodiversity, on the fisheries resource. It questions the capacity of the resource to be able to reproduce in new conditions, and so on. And this impact of climate change may also alter the ecosystems that are important for the fisheries resources, such as the mangrove and the nursery, once again. So if you have understood the ecosystem we have chosen is the mangrove and the nursery that depend on this mangrove. This is the area that is the object, it is the subject of our observatory. You can see here on the map Cayenne, that is the major town in French Guiana. And on this location, that is our observatory, we can see various uses, various stakes. For example, here you have the growing population with more urbanization that may have impact on the hydrographic basin that conducts here to the river of Cayenne. There is a bridge and the power station that are under construction here. You have recreational and professional fishing here, and also two fishing harbors located here. There are conservation areas. Well, there are many stakes. It's not exhaustive, but that's why we have chosen this ecosystem in terms of observatory and to monitor the various components and to have some indicators in terms of outputs of the observatory. So the action that we carried out are, of course, to collect samples on the different components of the ecosystem, about the mangrove, the tides, currents, and on the ecology of larvae of fish and shrimps. We also have implemented socioeconomic and anthropological surveys to better characterize the uses and the perception of the actors in this area. We have also organized participatory workshop to involve the stakeholders and the decision makers. And the last workshop was in October, and we have then collected their stakes and what are the next decision, what are the socioeconomic stakes in the area. So these are some indicators, some outputs of this observatory in terms of ecology. I don't want to go into the detail. I will finish by this slide to conclude that ecosystem-based management of mangrove is contribute not only to more sustainable fisheries, but it contributes also to more resilient fisheries facing climate change. Actually, ecosystem-based management is considered as a nature-based solution to address societal challenges such as climate change and mitigation of climate change adaptation. So I won't go into the detail, and I keep this story for the next meeting of the GCFI next year. So thank you for attention.
Thank you, Fabien. Maybe one question, a quick one, if anyone. No?
Thank you.
Okay.
Thank you. You know, French Guiana's European Department overseas has a lot of means to do a lot of good work and a lot of new collaborations. What is the current state of fisheries and the resources that they exploit? Are they doing well? Are they doing bad? What's the status?
Well, if I stay on the topics of this presentation and the concerned ecosystem with nurseries, we are concerned by two resources, the shrimp resource and the coastal fish. So for the shrimp resources, we don't know really what is the state of the stock, and there are some collaborative work in those days with the Fisheries Committee, and we have survey at sea to better assess the state of the stock. I hope we will have some results in 1 or 2 years. And about the coastal fish resource, especially the akupa, Cynosion akupa, we think it's overexploited, but especially because of the IUU fishing. French Guiana has to face this very important IUU fishing coming from Brazil and coming from Suriname. We have just finished an assessment also with WWF and the Comité des Pêches, and we have shown that the fishing pressure from IUU fishing has increased by 2 since 2012. So the situation is not good, and the stock, of course, is questioned and probably overexploited because of this.
Thank you, Fabien. I was also wondering, but we can have, we're going to move to the next person, but I was also wondering what indicators showed you already that the mangrove was in good health there? If you have a quick answer, otherwise we will...
Well, I don't know exactly which indicators I use to be able to say that the state is good, because it is the work of my colleague, a specialist in mangrove. We could have a discussion with them, and we are implementing now a new program that is called Magellan, and based on mangrove survey. So there are many surveys of the mangrove in the concerned areas that show that the state is quite good, but I am not able to say exactly what are the indicators to say this.
Thank you, Fabien, for the great presentation. Okay. So now we're going to have Sita Narayanan from the Guadeloupe Port Authority. And she's going to talk about monitoring and management of marine and terrestrial ecosystem in Guadeloupe.
Good afternoon, everyone. Before we dive into today's topic, I had to mention that I'm filling in for my colleague, Jennifer Tockney, who couldn't be here today. I'm happy to be with you, and I will do my best to share her work. Our focus will be on the progress we have made in monitoring our vital ecosystems. First, let me introduce you to Guadeloupe Port Authority. We don't just manage maritime access and port facilities, we also have the critical task of managing our natural areas. Our operations span 5 multi-purpose ports throughout the archipelago, and our numbers regarding commercial traffic and passengers demonstrate our capacity and reach. They also remind us of the impact our activities can have on the environment and why our commitment to sustainable practices is so important. The challenges we have faced over the last 15 years often required innovative solutions that didn't exist or didn't meet our specific needs. This situation led us to create the Coyote program. This program has become the flagship of the Port Authority's biodiversity conservation efforts and consists of 5 focused sections that guide our activities. Monitoring natural areas is one of them. Our main objectives are simple but essential to assess the cumulative impact of port activities on biodiversity and overall ecosystem health. By monitoring target species, resources, and ecological processes over time, we can ensure that our environmentally friendly actions are delivering real benefits. This monitoring helps us to adapt our strategies and ensure that we are not only meeting the current challenges, but also the future ones. So, the coming results highlight a 3-year monitoring study conducted by Créocéan across 12 sites in 3 ecosystems: coral reefs, seagrass meadows, and anthropized habitats you can see on the map. Over this period, 33 parameters were tracked to deepen our understanding of these ecosystems' diversity and changes. These parameters encompass both habitat structure and associated fauna. The study also focused on fish communities, specifically tracking the abundance and size of 61 species. Our findings on coral reefs are troubling. The health of these reefs is deteriorating, characterized by high algal cover and reduced diversity and density. This decline is largely due to the outbreak of stony coral tissue loss disease in 2021 and extreme bleaching events triggered by heat waves in the summer of 2023. In addition, a disease affecting sea urchins, particularly the diadem sea urchin, had led to further declines. These urchins play a critical role in grazing on algae, so their loss has allowed algae to proliferate, further stressing reefs. Despite these challenges, there is a glimmer of hope at the Mouchoir Carré station, which shows a stable or even positive trend. For fish population on coral reefs, identifying a clear trend remains challenging, as long-term monitoring is needed. Over the past 3 years, no major changes have been observed despite reef degradation. Fish diversity has remained steady, with 15 to 30 species recorded at each station, and overall abundance has been stable, particularly high at Folens. All trophic groups are well represented, reflecting a balanced community structure, and the slight loss of biomass at Mouchoir Carré and Frégate is attributed to larger fish observed in 2022, which were not seen in subsequent years. Lastly, the invasive lionfish has been observed at all stations, while the brittle star Ophiotella mirabilis, often found on Gorgonian fans, was recorded at 3 stations. In contrast to coral reefs, the monitored seagrass meadows show growth trends in structural parameters. The density and length of Thalassia testudinum leaves have increased constantly across all stations over the past 3 years, highlighting the effectiveness of conservation efforts we have made for this habitat. However, the stations at Petit Cul-de-Sac Marin are experiencing rising algal growth, which may hinder seagrass expansion. This could be linked to poor water quality or scarcity of grazing species. The invasive Allophila stipulacea was observed at all stations, with notable abundance at Folens. Fortunately, it appears to have stabilized and is coexisting with native species, as it has not expanded year over year. Seagrass beds host the lowest fish diversity among habitats, with a maximum of 6 species recorded. Fish abundance is also generally low, except in Folens, where a large shoal was observed in 23. Most of the fish observed were juveniles, underscoring the nursery role of these meadows. The trophic structure is primarily herbivorous, with these fish using the meadows as a feeding ground. Additionally, a noticeable increase in conch density was recorded at Folens from 23 to 24, likely linked to the meadow densification and increased leaf growth. In our anthropized habitats, the study of fish population at the Folens and Basse-Terre docks, as well as the Jarry breakwater, reveal high abundance and fish diversity levels comparable to those on coral reefs. In 22, the exceptionally high abundance and biomass recorded at Folens were due to a large fish shoal. The high fish population in these anthropized areas are supported by submerged structures which covered in rich benthic life, offer shelter and promote fish establishment. However, lionfish are also abundant in these habitats, posing a threat to native species. The current monitoring methods effectively provide a broad view of ecosystem health, but they have certain limitations. For seagrass meadows, structural parameters are sensitive to various pressures, but respond slowly to environmental changes. Adding physiological measures, such as nutrient levels in Thalassia leaves, would allow for earlier detection of disturbances and timely conservation actions. Coral reef monitoring methods offer valuable insights into coral cover, but are limited to linear transect. As possible improvement would be use of photogrammetry to gain a broader perspective and conduct more detailed analysis. To address the problem of underwater visual sensors for fish population affected by variability in fish movement and species different behaviors, we can think about video monitoring or environmental DNA. Given the slow growth of seagrass and coral species, ongoing long-term monitoring is essential to confirm current trends and assess ecosystem resilience. To strengthen the resilience of natural areas within the Port Authority, various conservation and restoration measures can be introduced. Installing more ecological mooring to reduce the impact of anchoring on seagrass beds, enforcing fishing restrictions to protect fish populations, conducting waste removal to enable natural species to reclaim their habitats, implementing artificial reefs, and enhancing sea urchins population by cultivating them in controlled environments and reintroducing them to the wild to curb algal growth, along with manual algae removal on impacted reef. Thank you very much. We remain committed to advancing sustainable practices that support both our natural environment and the communities that depend on it. Maybe I may not be able to answer the most technical questions, but please feel free to share them with us.
Thank you, Sita. Please, do we have questions for Sita? We have time. No? Thank you very much. Next we have Iker Irazabal from who is a program coordinator of the Dominican Reef Network.
Hi. Hello, everyone. My name is Iker. I'm representing the Dominican Reef Network. The Dominican Reef Network is an alliance of many different NGOs and government organizations in the Dominican that we all work to preserve our reefs. I'm here in representation of many different authors to talk a bit about a project that is very close to my heart and the challenges that we face. Okay, so what and why? As you can see, this is an island that is currently occupied by both the Dominican Republic and the Republic of Haiti. Sorry, I'm nervous as heck. It's the second largest island in the Antilles, and it's home to a very diverse host of reefs. We have the Atlantic coast on the north, that I'm going to be talking a bit more, and then we have the Caribbean side on the south. In 2019, stony coral tissue loss was detected in the north coast of the island. We can see here the spread that it had in those years. It was an emerging threat. And the National Disease Committee from the Dominican Reef Network created a plan for the study of coral disease as an emerging threat. This included a very detailed monitoring plan, something that has never been done before in the country, with a very structured design and experimental design. And the important thing is that it was sustainable throughout time and in space. So even though this plan is currently being done all around the country, most of the funding, most of the resources are in the southeast portion of the island, where Santo Domingo, the capital, and Punta Cana are. The north coast is very remote. There's very little support, no organizations currently north. So the Dominican Reef Network and its partners, we kind of adopted these sites. These are our sites from Punta Rusia, Sosúa, Terrenas, Galeras, and Samana. It's about a 300-kilometer coastline, so quite a big space. In each location, we had from 3 to 5 sites for a total of 20 sites, 3 transects, belt transects for benthic surveys, and each one is our operational unit. And importantly, we went there 3 times a year for 4 years to avoid pseudoreplication. So what is pseudoreplication? It's to incorrectly treat samples as independent, whether it's temporal, spatial, or by treatment, and it's something that has plagued research in the Dominican for many, many years. Because of lack of funding, usually you can only go 1 time a year, you can only visit 1 site, and you get the wrong conclusions from that. So how to avoid it? You do true replication, you have a proper model, you randomize your samples, and you have a clear strategy in mind. Master Yoda, actually, Master Croker down there in the audience says, no hypothesis leads to bad experimental design leads to pseudoreplication, and pseudoreplication leads to lack of understanding, usually by averaging. And we did not average, no. We have all the data here present. So every single column you see is the total count of colonies in every single transect. For brevity, we managed to identify to the species level, but here for brevity is just healthy colonies, sick colonies, pale, bleach, and recently dead colonies. So the higher you see the bars, more colonies were present, and as I said, there's every single transect for every single site for every single location. Bear with me for a second. As we can see, there was a minor mortality event in Sosúa in 2021 due to stony coral tissue loss disease in its final stages, but from 2021 to 2023, the reefs were pretty much stable. We had some very good outliers in Samana and Galeras, some specific sites with very high cover, but the reefs were pretty stable. So what happened in the middle of our doing this survey? Well, there's a graph showing thermal stress, and as we can see, the third quarter of 2023 showed massive thermal stress like nothing seen in previous years, and sadly, the prognostic is that we're going to experience the same ending this year. So what happened is that everything died, basically. We can see that thermal stress directly correlates with a major spike in bleaching and then high mortality across whole sites. It was pretty bad. What can multivariate statistics tell us? We can describe what happened, we can try to explain what happened. What we can see here is a non-parametric distance graph. Basically, all of these icons are every single transect as our unit for every single location for each quarter of each year. And they tell us a story. They tell us a story that long ago, the 4 nations or the 5 locations lived together in harmony. There is very little dispersion between samples, but then everything changed with bleaching. The variation that bleaching induced in the system was so great that it supersedes variations between species, between assemblages in the sites. And what's worse is that it never recovered. In 2024, even if we don't have as many dead or stay colonies, the composition of the ecosystem is completely changed from what was there before. We can see the years. We ran a permanova analysis. It's a permutational analysis of the variance. Basically, you run every single factor and its interactions and see which ones are relevant, and all of them are relevant, which is what you never want to see. But we can see that site and location is relevant, and location is relevant. So these reefs have a lot of independence, and even still, quarter, especially this quarter, was the highest damage ever done to these reefs. There's a bootstrap averages, and we can see again what I was saying. This is centroid of the data. There's all the sites previously independent of region, then bleaching, then post-bleaching. So what does this mean? We talk about data, we talk about species. I'm gonna say what this means to me, and if I cry, I'm sorry. This is me in 2018 in GCFI. I was fresh out of bachelor's, and I thought I had all the answers. And I was presenting about coastal erosion, about bleaching, but we haven't had a major bleaching event since 2011, so I thought we were in the clear. I left for my master's degree in 2019, and the last picture I took was the first report for stony coral tissue loss in the Dominican. Me and my friend did the report for AGRA, and this reef that was my favorite reef up in Monte Cristi when I returned from my master's degree was this. That's me crying in the picture. So the north coast of the island holds a very special place for me. What have we learned, and what can we do next? Well, we learned something that we have suspected for a while. There's a high independence among reefs. And when I mean reefs, I mean the sites within a specific location. Even within Punta Rusia, they behave differently. There's different assemblages of species, and they responded differently to both disease and bleaching. There's high intra-annual variability. If we only go there once a year, doesn't matter how many years we go there, we'll never see the full pictures. We need to go at least quarterly to be able to be just in time to see these changes to reefs. We've seen changes in communities, probably forever. We don't know what the future holds, but we're seeing the disappearance of species and changes in how these species interact in the reef. But the main thing is that collaboration is key. The major issue for Caribbean conservation is funding, both monetary, both personal. We don't have enough people to be everywhere all the time. And this project was only made possible thanks to interinstitutional cooperation. People from many different institutions that had their own personal projects, and they all decided to come together, put money, put effort, put time to visit this very difficult to access region for many years, and just to get data, just so we know what is happening. We need to expand efforts. My colleagues are going to talk about Monitorea. There's a project that we're starting in the Dominican Republic that aims to create the next generation of marine biologists and expand this to the rest of the to the coastline. And we need to monitor recovery. We need to still be in the water. And there was a talk about robots yesterday. I don't know if robots are the solution, but we need more people. We need people in the water, people that care, and we need to not give up. So yeah, I would like to thank all the people that participated in this expedition. They're the real heroes, and all the organizations both that funded this research or allowed me to be here to present this. And thanks to you for listening to me.
Thank you, Ike. I don't know if I pronounce it well.
Yeah,
Can you pronounce it?
Yeah, that's
Ike.
fine.
Okay. Any question for Ike?
No.
I have one.
Yeah.
Yeah, I was wondering what, because you ended by talking about not giving up. So I was just wondering how are you doing that? And
With great difficulty.
yeah, I can imagine. But like, you're still doing the research, right?
Yeah.
And you have planned to continue. So like, is there anything that has been maybe giving you some hope or being optimistic?
Yeah, definitely. We've seen reasons to hope. There's some sites in the North Coast that shows unusually high recruitment levels, even after bleaching, even after chronic tissue loss disease. We are working on those publications. And the reef is very resilient. This is worse than anything I've seen, but the reef is resilient. I'm hopeful for the people. I'm hopeful that we're still trying, so we gotta keep trying.
Thank you for doing that. We have one question. Sorry.
Yeah, I was, tragic. But I was wondering if you didn't talk about efforts towards transferring that into like the economic impacts. And you know, we saw this beautiful video last night about the new national parks and expansions of MPAs in your country. But it sounded like everything is great, but now you're telling us that it actually isn't that great. And you know, just so like, what support do you have from your government and communities?
Government support is limited because they don't have a lot of money to spend, and I don't think they really understand the need for long-term monitoring. One of the persons that contributed to this project is Jasmine, that she actually presented virtually. I think it was yesterday. She's a technician from the Ministry of Environment, and she's been really helpful. But on the macro scale, there's not a lot of support. Some of the areas here are technically within MPAs, but it makes no difference. We also do fish counts. I did not present this today. They're as bad as you expect, with a continuing decline in size. Diversity is still okay, but size classes continue to decrease every year. The communities are really helpful. We have a lot of support in all the different communities that we go. They know what we do. It's a small country. It's big, but it's small. So they know the 4 or 5 biologists that go out there every year. And there is support, but it's just not enough. We're facing threats. Like, we cannot stop bleaching. We cannot stop new disease. We can control fishing. We can somewhat influence coastal development, but it's hard because there's some things we just cannot control. We can try to adapt, try to know what's going on, and communicate that, and maybe, hopefully, the government eventually will decide to do something about it. Does that answer your question? Yeah.
So were you able to actually link the decline in fish size to the bleaching, or do we know if it's pressure?
There is fishing pressure. We're working on this is just a snippet. We're working on a way larger project. One of the leading theories is, well, how we're doing the fish counts to see, hey, if the reef is degrading, is this directly related to loss in fish habitat? But there is fishing pressure for sure.
Okay. Thank you.
Yeah, well, thank you.
So up next we have Louise Choureau, representing La Sommaire, an organization based in Martinique. And she's going to talk about coral reefs again. Maybe some hope in this one? Oh, no, actually. Spoiler. Not really.
Thank you, Frédéric. Bonsoir tout le monde. Buenas tardes. Good afternoon. My name is Louise, and I am currently working in La Sommaire NGO as scientific mission manager. And I would like to start this presentation by saying on the behalf of La Sommaire team that we are really honored to be here, and we are giving great thanks to GCFI organizer to let us the opportunity to present our work. It doesn't work. Okay. This work was carried out as part of the project Acropora, and I would like to thank all the partners who contributed to its outcome. As everyone knows, coral reefs are very important tropical ecosystem, as they provide habitat for about 25% of marine life and ecosystemic services. But these same ecosystems are under threat from multiple pressures. In response to the threat, conservation programs have been launched in recent years in the wider Caribbean region. The aim of these conservation programs is to boost coral population, particularly Acroporidae species. Despite their limited capacities in face of climate change and human pressure, these conservation efforts support the resilience of marine areas and the preservation of the species. I will briefly explain the context of this project because it's important to understand why and when this study research took place. Martinique Island is also affected by Acroporidae population decline, and this type of program was set up when Acropora cervicornis, known as staghorn corals, was rediscovered, and while it was considered to have disappeared from Martinique seawater. Today, this project is no longer ongoing due to the 2023 bleaching event, which lead to death all of the corals on the nurseries. In a nutshell, this program began in 2015 by collecting acroporas from Luca Havel site and implementing them in nurseries for propagation and transplantation on the reef. The main goals were awareness raising, communication, and in situ conservation as part of a marine area project in St. Luce. Many barriers made this project more complex. If conservation efforts were to continue, it was necessary to overcome the lack of data on the state and genetic diversities of corals in nurseries. So this is how Acropora project evolved from an operational in situ conservation project to a scientific research in collaboration with Borea Laboratory at the University of French Indies. For this new goal, 3 questions were to answer. 1, do nursery efforts using propagation techniques allow colony to develop? What is the genetic diversity in Acropora cervicornis remaining colonies in nurseries? The prevailing hypothesis, according to local experts, was that the nursery contained population of clones. And are Acropora cervicornis in Martinique genetically different from those in the Lesser Antilles? This genetic work was based on study carried out locally in order to be able to compare data sets, and I would particularly like to thank Aurélien Japot for his collaboration. The study area is located in St. Luce. It doesn't work. 2 nurseries have been set up on 3 different sites at about 10 meters deep. Monitoring are regularly carried out by different actors, and it suggests that these sites are under multiple anthropogenic pressures from both land and sea, but the intensity of those pressure varies from east, very high, to west, very low. For genetic analysis, 95 samples were taken from the nurseries and compared with the database resulting from Aurélien Japot's work. Corals were identified for both genetics and monitoring. Among the 95 samples for genetic, 10 corals of each site were monitored for growth and conditions, and 10 corals from each nursery as well. Monitoring was done every month. We have conducted measurements on selected corals to assess total linear extensions tissues in order to evaluate growth and productivity. For analysis genetics, we used microsatellite markers designed by Iliana Bohms, and then we conduct data analysis to assess distinct genotypes, genetic diversity, and distances. Monitoring allows us to present growth dynamics and productivity dynamics over 5 months. On graph A, you can see the growth according to the size of each colony, represented by blue squares. On graph B, you can see productivity according to the size of each colony. The results indicate that smaller corals have bigger productivity than bigger corals. This data was interesting to us as coral development seemed to differ from one site to another. This graph shows the average size of living tissue of the 10 corals monitored per nursery over 5 months. This result shows differences between sites. For example, it seems that coral on pink sites grew faster than those on green sites. But it also shows differences on the same site. For example, in the blue site, you can see different growth and dynamics between nursery. For genetic results, first of all, there are 7 distinct genotypes which have been detected in our samples. These results belie the hypothesis that the corals on the nurseries are only clones, although genetic diversity and richness have low value in our samples. To go a bit further, we wonder what might explain the difference in development among the corals, both monitored and genotypes. On this chart, we are looking at the average living tissue per genotypes, and we wanted to test if there is a potential influence of genotypes on growth. Results show that genotype 1 has a significant higher average growth than genotype 3. The influence of genotypes or environmental conditions on coral development remains to be explored, but those results show significant differences on growth dynamics amongst detected genotypes. To go a step further in the study, in which the main goal was to focus on coral conservation in Martinique, and because we have chosen the same methodology than Aurélien Japot's work, we could have a look on what is happening on a larger scale and make our humble contribution to the study of Acropora cervicornis genetics in the Caribbean. This result shows Acropora cervicornis genotypes from Guadeloupe, Saint Martin, and San Barthélemy, represented in white squared on this graphic, and Martinique Acropora cervicornis in red. As the one from Martinique are not a natural population, because they are from selective propagation in nurseries, I will not go further into population genetics, but it was interesting to see that there is polymorphism with several genotypes represented within the population. 3 groups are formed, and 2 of them are well clustered. In those groups, all identified genotypes are found at all sites, so in all populations. Now that I present the results, I will share some perspective on them. In our study, we integrate negative values for growth as we wanted to represent what was really happening on coral nurseries for conservation purposes. What we could observe is that in the nurseries, coral growth and productivity is quite low, but we obtain similar results for productivity dynamics. For genetics, analyses were made on corals which were propagated for several years, since 2015. And we know that there has been mortality among the stock. That could explain why there is low diversity. It would have been better to know genetics diversity to adapt collecting methods on mother colonies when the project started in order to maximize genetic diversity. Also, genetic diversity in the Caribbean needs to be studied on natural coral population. When comparing results from natural population with ours, we found out that reproduction was mostly asexual. It's logic when within a program restoration using propagation methodology. But it's hard to conclude as samplings is not made on natural population. But in 2023, we have been very pleased to witness coral spawning on nurseries. It was very emotional moments, and we hope that coral larvae would have set up somewhere on the reef. To give feedback about this project, though we have faced uncertain budgets, no clear initial goal and lack of political involvement with differing opinions between stakeholders about coral conservation, this project raised awareness and strengthened partnerships with local government, technical actors, university and research labs to humbly contribute to science. To finish, I would like to take the voice of corals and marine biodiversity to say that we must remember extinct species and to fight the ones that are in danger and to preserve all of the living. Thank you very much.
Thank you, Louise. I'm sure we have questions for Louise. Okay. Do you want to tell us a little bit about how you can explain or understand the lack of political involvement?
Thank you for this question. This is a tough question. So I would say that this project could have put on the table the problem of corals, but the problem of all marine ecosystems, which we don't usually see when we look at the sea. We see it's blue and beautiful, and we don't question what is underwater. And I think most of the political structures maybe don't know about it or don't pay attention to that or have problems that they have to deal with before. So that put them far from the questions. I would say this is a first step. So raising awareness with this kind of project is very important to implicate citizens and make a change in the society.
Thank you, Louise. Up next is Henri Vallès, who is a senior lecturer in ecology at the University of the West Indies. I believe this is a pre-recorded one.
Frederic, I don't think it's pre-recorded.
Oh, what? Sorry?
I don't.
No, you hear also you're doing it in live. Okay, great. Hi, Henry.
Yeah.
Hope you're well. Thank you for joining us.
You guys hear me?
Okay, we have wait a minute. Okay. I think you're good to go. Thank you, Henry.
Okay, can you hear me? Yeah. Sorry. Can you guys hear me?
Yes, it's good.
Okay,
Well, we don't
so I
see
guess
you,
I'm
but
gonna
we
go.
see the PowerPoint, so it's good. Thanks.
You can hear me, right?
Yeah.
Yeah. Okay. Yeah. Okay. So hi everyone. Thank you for coming to this talk. I'm going to talk about a multi-institutional, multidisciplinary, and regional project funded by the Caribbean Biodiversity Fund, aimed at restoring the wave attenuation effect of Elcon coral stands in the Caribbean reefs, so as to help fight against coastal erosion. And before, I'm not sure, can I pass the slides? How do I?
How we pass this?
Do you,
Oh, also Fadila is doing it, so you can say next.
Oh, okay.
Next.
Okay, next
Say
slide.
next.
Yes, please. So before I talk more about the project, just let me give you a little bit of background. I'm sure that this draft, published by Garner et al. In 2003, needs no introduction. shows a steady drop in coral cover over time from the 70s to the early 2000s. And as we know, things have not improved since then. This graph also shows that the biggest drop in coral cover takes place in the late 70s, which was when acroporate corals were decimated regionally by the white band disease. So this graph supports that acroporate corals were a dominant feature of Caribbean coral reefs. Can you pass the slide, please? Next slide.
Yes, it's good.
And if you go back to the seminal work of Goro in the 1970s in Jamaica, Elgon corals at that time definitely dominated the shallow wave break zone of Jamaican fringing reefs, where their complex 3D structure likely helped attenuate incoming wave energy. Next slide. The assertion that Elgon coral stands on the break zone can significantly reduce incoming wave energy is supported by modeling studies like the one by Rosario Cano here shown and colleagues, who compare simulated wave dynamics between reefs with and without Elgon coral stands in the break zone, and show that, next slide, and show that having Elgon coral on this reef zone contribute to a wave height attenuation of more than 75% under moderate to extreme wave conditions, and to an overall substantial decrease of wave energy reaching the shoreline. This is important because coastal erosion, due to increased intensity in wave energy, is a major concern for small island states in the face of climate change. Next slide. And this is a concern with strong economic implications. For example, in Barbados, the average annual coastal erosion is estimated to be 0.3 to 0.4 meters per year, which is very worrisome. And the government of Barbados, which is, by the way, where I'm based, has invested about $20 million between 2002 and 2010 in great infrastructures, such as the breakwaters that you can see on the bottom pictures to the right, to address this issue of coastal erosion, and this is becoming an increasingly frequent sight on Barbados' west and south coast. Next slide. At the same time, over the last 2 decades, there have been multiple reports across the region that Elgon coral is coming back naturally. And this was also noted in Barbados in 2016, with the first mapping of Elgon coral colonies along the west coast of Barbados, and the identification of sites with high abundance of this coral. Next slide. So, over this background, I would like now to introduce the overall objectives of the OneSec project. The first one is, as we have deduced, to restore the stance of Elgon coral, to restore the stance of this Elgon coral on the wave break zones of selected fringing reefs. And to do so, the project is adopting the techniques developed by Andrew Ross from Seascape Caribbean, a Jamaica-based firm that specializes in the restoration of Elgon coral. Next slide. Moreover, this expected enhancement in 3D structure through Elgon coral restoration and its effects on resident organisms and in the benthic community will be periodically documented using photogrammetry and standard benthic and fish surveys with the support of Duatec, which is a firm specialized in photogrammetry and reef monitoring. Next slide. The next objective is to show that this enhanced reef physical structure actually reduces vulnerability to coastal erosion. And to do so, we will be monitoring the physical wave environment at the outplanted sites to generate data for coastal risk modeling. And this exercise specifically will be conducted by a team led by Mike Beck, based at the University of California in Santa Cruz. Next slide. The final main objective is to build local stakeholder capacity to implement reef-scale Elgon coral restoration. And this will be achieved by engaging and training local key stakeholders and by assessing the impact of the project on local communities via socioeconomic surveys. These stakeholder activities will be conducted by the University of the West Indies in Jamaica and Barbados. I am, by the way, based at the UE, at the University of the West Indies in Barbados, and by the Antigua and Barbuda National Park Authority in Antigua, who are also themselves benefiting from the training. Next slide. Importantly, all this will be replicated on 3 different islands. So this is happening at the same time in Jamaica, in Antigua, and Barbados, using the same methods and techniques to assess transferability and replicability of the approach. And this should ultimately provide valuable lessons, knowledge, and experiences, which will then be used to develop a toolkit and a policy guide for Elgon coral restoration for wave attenuation in the region. Next slide. So I will now provide some more detail on some of the key project activities, and I will also summarize some of the milestones achieved in Barbados, which is where I'm based. So essentially, in terms of selecting donor material, we seek to have a mixture of 4 to 5 different donor colonies, likely belonging to different lineages. And you can see from the picture there, a process of taking a piece of a donor colony, these colonies, before selecting them, should look healthy and fully recovered from last year's regional bleaching event. And we have actually been fortunate in Barbados that most of our Elcon coral has recovered after bleaching last year. We've seen relatively low levels of colony mortality. We then collect the fragments from each colony. In total, and the fragment that we collect from a given colony is less than 10 to 15% of a colony, and turn those fragments into hundreds of small nubbins that are approximately the size of a thumbnail. As you can see there, which are attached then to a nursery frame using a line and using specialized cable ties to secure them onto the line. And they're left there to recover for 100+ days. You can see to your right, a diver setting up the nubbins onto the line in the nursery, and you can see what a nubbin looks like approximately 3 months after having been collected and secured onto that nursery line. Next slide. The idea is then to outplant them at high density, that is about 1 nubbin per meter square, within a gridded permanent quadrat the size of approximately 2 Olympic-sized pools, so a 50-meter by 50-meter plot at less than 2 meters in depth. And what you have here is a diagram showing the potential location of one such plot on a Barbadian fringing reef. The picture above to the right is a recent picture of the same reef showing 4 breakwaters placed there to attenuate wave energy and to reduce coastal erosion. And this is how we are actually currently addressing this problem in Barbados, by setting up more and more breakwaters. So the idea of this project really is to try to find a nature-based solution that can act as an alternative to just putting these artificial structures on the reef. Next slide. We also follow a before-after control impact design with biophysical data and regular monitoring being taking place at 3 sites before the outplanting, 1 site left as control, and 2 sites assigned for outplanting. And then, importantly, this is again replicated at the other 2 islands. So reminder, we're doing this almost simultaneously in Barbados, in Antigua, and in Jamaica. Next slide. So that just shows basically the location of the other three countries that are participating in this, and ultimately the idea of replicating this across 3 different islands is to assess the extent to which this approach is transferable across islands, and therefore replicable and potentially scalable, and to learn from those experiences and exchanges. Next slide. Yeah. Okay. Yeah. So we are actually, as I mentioned earlier, we are going to be collecting biophysical data. Me and my dad, we're going to be collecting data that include photomosaics at the resolution of the entire plot. So the 50 by 50 meter plot broken down in smaller quadrats, and then creating photomosaics that will be ultimately patched together. We're also conducting agra-type benthic and fish surveys at predetermined time periods, and we are deploying wave energy, temperature, and salinity sensors just to get a sense of the physical environment surrounding the outplanting sites and the control site, as well as in the nursery. Next slide. So I'm going to speak now in terms of what we have been doing in Barbados specifically. The funding for the project arrived in February 2024, and since then we have had multiple events of stakeholder outreach, engagement, and training, and this is a very important component of the project. So we had an inception workshop in April 2024, where a diverse slate of stakeholders participated in the selection of the sites designated for outplanting. We have also had multiple focus meetings with fishers and surfers, which are 2 of the primary key stakeholders that should have a say in terms of where this activity should take place. Particularly with the surfers, we had several meetings, so we're quite interested, but also had some concerns about which reefs we will be using for our outplanting sites, as in terms of how that could actually interfere with the surfing dynamics that exist currently in Barbados. And we have also had several days of volunteers participating in the creation of the nubbins, the fragmentation of the bigger pieces into smaller pieces, and in the population of the nursery. Next slide. So just to give you a sense, in Barbados we have identified 5 different donor colonies, which we have called different colors or labeled as different colors. 2 different sites likely belonging to 5 different lineages. And fragments from these colonies have been used to generate approximately 3,500 plus nubbins, small nubbins, again, the size of a thumbnail. Next slide. And then these nubbins are transplanted to the nursery. This was done in May 2024. Picture to the right gives you an idea of what the nursery looks like. And at the same time, we have been doing weekly monitoring of the nursery and quantifying nubbin mortality over several months, and explore the role of nubbin size, because there's quite, even though I'm referring to, thumbnail size, there's variation in terms of the fragments that you get. So we've been walking around looking at how that influences nubbin mortality going forward, and also looking at colony lineage in remnant mortality. And what we found basically is that nubbin mortality levels were overall low, and depending on both nubbin size, the larger nubbins showing greater survivorship, and also depending on colony lineage. With the red lineage here to your right in the graph, you can see that arrow bar showing the lowest mortality, and the white one showing the highest mortality. Next slide. We also found that nubbin growth rates that differ among lineages, with the white lineage ranking high in growth rates. So interesting enough, even though the white lineage was the one that appeared to have exhibit higher mortality than the others in terms of when it was fragmented and left to recover at the nursery, it actually is the lineage that exhibited some of the highest growth. So similarly having a trade-off there. Next slide. However, we were expected to start our outplanting in October 2024, so just last month, but we have had to face the reality of an increasingly hostile natural environment. First with the passage of Beryl, which had a very strong impact in Barbados, particularly on the south coast, but also on the west coast. Even though it did not hit the island directly, and it was completely unexpected, but it has done, it did quite a bit of damage to the island infrastructure, and also changed the landscape of the Beti communities on the south coast and partially at the west coast as well. So what I'm showing here to the left, you can see a picture of some of the colonies that we had at one of the donor sites in the south coast before Beryl and after Beryl. Next slide. We've actually, at that donor site, we've quantified 100% colony mortality. And this was a relatively healthy population that have actually managed to recover from last year's bleaching episode. And we have another site on the west coast where we experienced about 45% colony mortality. So basically, colonies were swept off there on the ground and turned into rubble. And next slide. And as you can probably deduce, we are currently experiencing a massive heat wave, even more important than last year's, which last year was already a bad one. And now we are seeing substantial bleaching at the nursery in terms of the nubbins, but also we're seeing quite a bit of bleaching at the original donor colony populations at the donor sites. Next slide. So what's next for us? Well, so I just want to say before going to these slides, actually, that in light of the massive bleaching that we're seeing, we're not outplanting as we had planned in October. We are going to wait it out and see how the nubbins in the nursery recover. If they recover, we are seeing high levels of mortality. Interestingly enough, again, the white lineage, which was the one that showed higher mortality rates during the fragmentation process, but higher growth, is also showing lower mortality during this bleaching event. So maybe something about that one specifically that may be worth looking into in more detail. So we are at this stage now simply tracking the fate of the nubbins. Next slide. Moving forward, so the next slide, in that direction. Identify new potential donor colonies with the highest heat resistance. So there are currently some students that are actually doing surveys along the west coast at peak bleaching to see whether we can identify donor colonies that show high heat resistance, as they could be potentially used in the future. Next slide. We are, as I said earlier, waiting until the temperature goes down and then show signs of recovery before outplanting. Next slide. And then we are monitoring both the nursery and parental populations, but also environmental factors. We are We plan to deploy the wave energy sensors very, very soon. We're collecting data in salinity and temperature. We're also looking at water quality data, like nutrient data. And we are also collecting tissue samples for DNA analysis just to confirm lineage identity. Next slide. And everything that we have done so far is helping us refine our methods further. Next slide. And we are at the stage also continuing to talk about what we're doing and engage stakeholders and continue to train stakeholders in various techniques that we're using. Next slide. And that's about it. I'd like to thank some of our local partners in Barbados, specifically the National Conservation Commission, Coastal Zone Management Unit, and Coral, an NGO that deals with coral restoration. And I'm happy to take any questions.
Thank you, Henry. Definitely sad to see the images after burial. We don't really have time for questions unless there is a burning one. Otherwise, you can write it directly on the app. Is it? Oh, okay. Yeah. Thank you, Henry. Good to see you.
Bye. Thank you.
So next we have Aldo Croca, who is a Marine Conservation Program Manager for the Central Caribbean at TNC, the Nature Conservancy.
Hello, everyone. So I am Aldo. I am the Marine Conservation Program Manager for TNC. I have been working for the organization for the past 5 years, and today I came to tell the story of our monitoring program called Monitorea, and tell you how this project has been a roadmap to monitor mangrove, seagrass, and coral reefs. And I'm going to show you some of the preliminary results that we have during the training sessions that we have performed and during the first pilot project that we conducted. So Monitorea is a project that is originated from a strategic alliance between the French Embassy in the Dominican Republic, Fundemar as the leading institution, the Ministry of Environment in the Dominican Republic, and the Nature Conservancy, who has been providing the technical expertise and support for our partners to develop the methodologies and the objectives that this project entails. So what is a good monitoring program? The answer is simple, but in terms of budgeting is complex. A monitoring program that is good is one that is able to split the natural variation that you normally expect in the environment from variation that is introduced by human activities. So that means that a good monitoring protocol or monitoring program should be replicated properly in space and time, and Iker show a good example of what that entails. So the general objective of the Monitorea project is to propose a methodological framework based on cumulative knowledge for the integrative monitoring of health status and restoration of coastal marine ecosystems. So the project revives the CARICOM, Caribbean Coastal Marine Productivity Project, a network that understood the value of monitoring the three main coastal marine ecosystems that we have in the Caribbean: mangroves, corals, and seagrasses. And they put a value in understanding the natural connectivity that we have in these ecosystems. This project has three major work packages. The first one is the harmonization of a structure of coral reef ecosystem monitoring. The second one is the strengthening of scientific research. And the last one is the dissemination and awareness. Today, I will be focused on the first work package. As you may know, a monitoring program needs to conduct pilot studies. These studies are important for us to understand whether or not the methodology that we are proposing is feasible in space and time, whether or not some of the methods that we propose needs to be revised or confirmed in the field, because sometimes you put something in the paper, but it might not be a feasible solution or a feasible approach. So we conduct a pilot field studies to test the methodologies that we were proposing for the three ecosystems, for mangroves, for seagrass, and for coral reefs. And we did that because we understand that data collection requires and demands exhaustive training. So far, we have completed 9 trainings. We have trained 72 people. 58% of them are women. 11 institutions have been involved. And right now we are ready to implement one national monitoring that will, cover 15 locations across the island. And we have signed 3 research cooperation agreements with national and international universities to ensure that this monitoring program will continue in time. So today, my aim is to present a summary of results from that pilot study. And the location that we selected was Valladolid, and we did this during this year. I have highlighted some of the indicators that we collected information in this pilot study on mangrove, seagrass, and corals. And again, this is not something to show what are the trends in these sites. It's rather to show you what is the roadmap that we have followed to implement this project. The mangrove protocol is inspired in the CARICOM protocol. The seagrass, we use for the seagrass the CARICOM, AGRA, and CARICAS. And finally, for corals, we are combining the GCRMN biophysical monitoring protocol and the AGRA protocol. That means that this project is not reinventing the wheel. We are taking the advantages of well-known and successful and widespread monitoring networks for each of these ecosystems. We have took their lessons, their success, to be implemented in the Dominican Republic. So for mangroves, the philosophy of our monitoring program is to replicate locations at the level of sites. So for the mangrove, we selected Palmilla. We have 3 sites for this location. And in each site, we have 3 plots, 10 meters by 10 meters. And in each of these sites, we monitor individual plants and also juveniles to get some information like indicators of the health status of this mangrove ecosystem. Here are some examples. The first figure shows you the cover of mangrove in square meters. As you can see, there is a lot of variability within that location. And then we show also the standing biomass in grams per square meter. And again, you see a lot of variation. If you see the size structure based on determination or estimations of size of heights, you will always see there is a lot of variation in terms of size structure. So what is the message of this? Each site within that location tells its own story. Therefore, averaging these sites to tell how this location works or is behaving is not a good approach. We also do some drone mapping for our study area. So you can see we can generate 3D models and orthomosaics to see like elevation models to see if how heterogeneous is the mangrove at that site. Following the same logic, in Palmilla, which is the same location where we had the mangroves, we selected 3 seagrass beds. And for these, for these sites, we estimated the shoot density for Thalassia, Syringodio, Alodule, and other seagrasses. And then the standing biomass in grams per square meters. You see the above ground portion of the seagrasses with the larger contribution for the non-photosynthetic part, and then the below ground to the right. Each bar corresponds to a single site in that location. Again, a lot of variability. We also conducted productivity. As you can see, the average ranges from 3.78 to 2.23, and also the turnover rate that varies from 143 days to 154 days. So again, a lot of variation at the scale of site for that location. So averaging might not be the wiser strategy. In terms of coral reefs, here I show for 3 sites in that location that is very close to the seagrass and the mangroves. You see the live coral cover in average, and we are showing here only live coral cover in yellow, in orange, and macroalgae in green. So the highest coral cover is found in Peñón site. Sorry, the largest macroalgal cover is found at Peñón site, and live coral cover does not exceed 5% at all sites. But again, there is a lot of variation in terms of macroalgae cover at the scale of site. To the right, we have the average recruits per square meter. Again, a lot of variation depending on the site. And to the bottom right, we have the average biomass of fish, accounting for herbivores, commercial, and others. Again, a lot of variation. So what are the next steps? Right now, we want to replicate this at the national level, following the same hierarchical design that Iker explained before. It's a hierarchical design where we will have 5 different sub-regions in the country: northeast, northwest, east, southeast, and southwest. And in each of these sites, we will have locations. And for each of the locations, we will have sites. So we will have a full understanding of spatial variation across the island. And that variation is the baseline for us, and is going to be taken as normal variation. So we will be hopefully able to separate that variation from human or disturbances introduced variation as we move in time with our monitoring program. So these are some of the photos of the training. It's very laborious. A lot of people have been trained. By the time I'm talking here, some of these guys are already in the field collecting data in some of the locations that we have that I have shown in this presentation. This kind of effort is only possible with the contribution of multiple NGOs and multiple stakeholders. Here we have people that come from the academia, but we have also trained technicians that work in their local communities. And the beauty of this project is that we have managed to engage these people so they understand the reason why they are monitoring, and they understand the value of monitoring for them to preserve their ecosystems. With that, I would like to finish my presentation, and I am happy to take questions. Thank you.
Thank you, Aldo. Any questions? Okay, we have 1 minute, so I may have one, please. No, I was wondering what is the main challenge to work at the seascape level and to integrate the 3 types of ecosystem, which we know is important, but at the same time, I imagine that it's probably difficult. So I was wondering if it was people, government, or technology lack that would be the main issue you have faced.
So in the Dominican Republic, bringing together different stakeholders have not been a limiting factor. Everybody is convinced and aware of the importance and the relevance of this project. I believe that training has been a key. We have managed to invite and to pay for the participation of different people, and they have been benefited from these trainings. I believe that the major challenge for these kind of programs is to keep them in time, to be sure that this project is going to last and is going to produce the time series that we need to understand what is normal from what isn't normal, right? But I believe that since we have seen the route with the local people, by the end of the day, we will manage to find the funding, and we will have the human capacity, the labor and the engagement of the people for this program to continue.
Okay, thank you, Aldo. I look forward to continuing this conversation over the break. But before that, we have our last speaker, who is Cléo Baron, Terrestrial Environmental Project Manager of the Guadeloupe Port Authority. And she's going to talk about mangroves again.
Hi, everyone. Nice to meet you. Buenas tardes. Bonjour tout le monde. Donc I am Cléo Baron, sorry. I am an environmental officer at the Guadeloupe Port Authority. As Sita Narayanan said before, the Port Authority of Guadeloupe is working and managing its own natural areas by implementing nature-based solutions. And today I'm going to show you what we are doing about mangroves. At the Port Authority of Guadeloupe, we have a volunteer project that is called Life Adapt Island Project. It's a 6-year project that started in 2019, and it will end next year. It's a 4.7 million euros, financed half by European Commission and half by the Port Authority. And we have 2 partners, Pilot for Dev located in Brussels and Eurapeg located in Guadeloupe. The 4 goals of this project is developing an innovative and replicable adaptation strategy, restoring physical connection between the major coastal ecosystems, so coral reefs, seabeds, and mangroves. It's also involving civil society and communities and replicating those innovative techniques in the Caribbean. So the Life Adapt Island project is using cross-cutting approaches. We are working on our socio-economic impacts, for example, with ecotourism, and we are also looking for our economic impacts. And maybe you were there yesterday night for the Cinefish and you saw our videos, but we are also raising awareness with those kind of videos, and we also have a website and an Instagram page. The QR code will be at the end of the presentation, don't worry. So as I said, we are working on the marine ecosystem also, first for seagrass beds. The objectives for seagrass beds is to reduce the anthropogenic pressures, to raise awareness, because seagrass beds are not very known for the population. And we are also testing some prototypes. To do that, we create eco-moorings and screw anchors. We also create an underwater trail in Marie-Galante. And we are doing Thalassia testudinum experimentation with fruit collections, in vitro germination for plantation of plants. About coral reefs, the objectives are to maintain a sufficient biological material levels, to help restoration, of course, and to acquire data on spawning, also to increase both the genetical diversity and the potential of success rates. To do that, about corals, we have 2 farms with 17 trees, 2 domes, 2 tables, and some cuttings. We are doing annual transportation operations with Acropora cervicornis and Acropora palmata. Also, we are doing annual coral spawning observations and collations. And we are struggling against major bleaching events. Yeah, that's all. So going back to the mangroves, so first I'm going to let you see where are the 2 sites we are working on, the land-based sites. They are located in the center of the island, in the very urbanized zone that used to be actually a mangrove and the wetland area. We have 2 restoration sites. The first one is the more little one, where we have trait invasive alien species, and we also created a 450 meter square pound. We also planted some red mangrove on this site, and the second site is a bigger one. We do the same work. We didn't dig a pound, but it's a bigger site, and we planted white mangrove. So, as I said, to understand why we chose these 2 sites, the Jarry area is a very urbanized zone because a lot of commercial buildings were built, yeah, and that's why we lost natural area. And also, when it's raining a lot, it's a flooding area. Unfortunately, those 2 sites are easily to access, so we have to struggle against embankment, lots of embankment, with all type of things. We can find trucks, cars, wash machines, and also little waste. All those embankments made the soil very dry, that mangrove can't grow on those sites. We also have to struggle against pollution. We have a little canal going next to those sites, and we have pollution with wild dumps and plastic waste. And we have this canal and the marine pollution. You can see on the picture. We also have to fight against invasive alien species. Those species are growing on dry area, so embankment made the soil dry, then the invasive alien species can grow. The objectives for the mangrove work is to restore this natural area, the wetland habitat for mangrove and for all the species, fauna and flora that can grow. We want to allow the development of mangrove in a flood-prone area. And we also wanted to diversify mangrove species. To do that, we have several actions. We excavate some soil, we manage the invasive alien species, and we are planting some seeds. So as you can see on the picture at the top of the presentation, in 2020, the little site was green, but it's actually invasive alien species. So the first walk was to put them away. And then we dig on this site, and the other site, we dig to find again the sea level, or close to the sea level, so the water can come from the soil and from the sea and from the canal, so we can have really a wet area. In this little site, we decided to create a pond, so we can have more water. From 2020 to 2024, you can see that the site is more wet, so that's good news. On this little site, we also decided to build a nursery. We have 3 structures on the pond that can storage 300 plants. So we will put mangrove species, of course, but maybe we will try to have some other plants for wild forest, for example. On all of the sites, we planted red and white mangroves, but we also have other plants that we are going to plant soon. For this project, we also monitor, we are doing a monitoring to see how the plants we are planting are growing. So we are following the mortality, the states of health, also for the leaves, the presence of fruits or flowers, the predation also. So if you want the results, they are on our website also, the Life Adaptable Island project. Actually, it depends on where the plants were planted. But when they are growing very well. And also the good results about mangrove, and that's why it's really nice to work with mangrove, is it's actually species that are growing easily when they have a little bit of water, because the water in this area is not very salty, but mangroves are growing. And we also have a lot of natural growth that is very good. We also have fauna that is going on this site, so we are happy about mangrove, because the results are not the same as we see with everybody for corals and seagrass. I think that's all. So thank you everyone for listening. If you have any question, I'm here. Don't forget to visit our website. We also have a newsletter if you want to see, and the Port Authority of Guadeloupe has a YouTube page where we have some videos if you want to look. Yeah, thank you.
Thank you, Cleo. We have time for questions. Yes, Lindy.
Hi, good afternoon. I have two quick questions. First one, as a non-European Union, is the size of the restoration sites 45,000 square meters, or was it a different size, a smaller size?
I think I didn't understand.
On the second or third slide you had about the restoration areas, you had two values.
Yes.
And I wanted to know is it the 47,000 square meters?
Yeah.
Yeah?
Yeah.
So these are big areas?
Yeah, it's a big area.
Okay.
Yeah.
And then my second question was in your distribution of plants at the end. You had lots of red, lots of white, mix of the dragon's blood or whatever it's called, and very small amount of black mangroves.
Yeah.
Is it because of the distribution of what's there currently, or are you trying to replicate what is naturally found on Guadeloupe in other areas?
So we wanted to have red mangrove in the little sites because we didn't find them around the site. So we wanted to try to diversify. We wanted to test if it's growing on this site. So we have more white mangrove. That's what we decided to put some red. And on the other side, actually, it's, we saw that it's easier to put white mangrove, that it's growing easier. So then we decided to put white mangrove.
Okay.
Okay.
All right, thank you.
Okay.
Yes, thank you. And just to conclude, any more questions? Because I was wondering with, I mean, it could be also Sita, your colleague, I don't know if she's still here, who could answer. But the idea behind like restoring these areas, when you showed the threats, I didn't see anything about like the port itself. So I was wondering, is it a mitigation action to compensate the port threats as well, or it's not?
Actually, for example, we have construction in this moment, and we are doing some compensation works. But we have two types of works. We have volunteer actions like this, and we also have compensations. We have several measures. I don't know the English names, but we have following, monitoring, and compensate. It's like, I don't know the English terms, but we have to do that for the state. It's obligatory.
You can say in French. Yeah.
Donc on a des mesures ERCS. I don't know the name in English for the building we are doing, but we also have some volunteer actions.
Okay, so they are totally separate.
Yeah, we are doing quite sometimes the same thing, but we have, for example, for the compulsory measures, we have some stuff for birds that we are not doing in the project, for example. So it can be completely different.
Okay. Well, thank you very much. It's exactly 4 p.m. Well, minus 1 minute, okay. But I will conclude the session. Thank you, Cleo. And thank you everyone for engaging in this session. Although we saw some depressing news, I hope we can, well, actually keep hope after this and continue working together, because as we've seen, collaboration is key, but also bottom-up approaches are essential for this work to be well implemented. So thank you everyone, and you can have a nice break. But before, Kim has a question. I didn't catch that. I'm sorry.
Wants everyone to go on the dock for a selfie. So
okay.
Can you repeat it on the mic?
So everyone, after you leave this room, you can go on the deck.
The dock?
Or the dock.
Yeah.
Yes. You can go on the dock to have a selfie picture. But before all the presenters of this session, it would be nice to have a little ecosystem and habitats picture. And yes. Yeah. And also our shared ocean grantees. I don't know all of you, but you probably know who you are. Yes? Okay. Yeah. But I know there is Estela, Jamie. It's good? Yeah. We all know? Okay. Because I'm one. So this is why I'm like, okay. Okay, everyone. So outside a selfie, and for the presenters and no. Kim said no. Okay. Okay, in 15 minutes, please gather outside on the dock for a selfie. But in the meantime, you can enjoy your break. And for those who presented, please come in front. Yes. And the grantees as well. Thank you.